Hello there, Dr. DiMaio again. This is part two of your immune system. Part one was the innate non-specific immunity. And this one right here is going to be, sorry about the mic wasn't close to me. This is part two of your immune system. Part one was innate or non-specific. This is part two, meaning the specific immunity. And we have these two main parts of our immune system. One is non-specific, things that happen on the surfaces of your body and in between tissues. Not that this doesn't happen in between tissues, the adaptive, but the adaptive is more specific. It's called specific immunity because it's the creation of specific targets and finding the target and going for them. So we're going to create some cells that are going to be programmed, so to speak, from APCs. An APC is an antigen presenting cell. And we'll talk about what an antigen is and what an APC is and how it works together. We'll talk about how we get T cells that are going to be programmed to fight specific antigens or bacteria or viruses. And we'll talk about how they make clones of themselves and also how they have memory cells. Not only T cells, there's basically two types of T cells we'll be discussing, the T helper cells and the T killer cells, or cytotoxic T cells. And they have a protein on it. It's identified by this CD4 for the helper cell, and CD8 is the killer or cytotoxic cell. And then we have B cells, and B cells will actually form into plasma cells and create antibodies. And we're making specific antibodies for specific antigens, such as viruses, bacteria, and even cancer cells. So here we're looking at the adaptive or specific immunity. There's two basic types. We have cell-mediated, which is like a cell attacking other cells, or cell-to-cell combat. And then we have humoral or antibody mediated, and that has to do with B cells and the production of antibodies. So the thing that makes this unique is that we compare and contrast the innate immune system. If you look at this right here, let me see if I can get my little pen. So this is called the innate immunity, this box right here. And then this is called the adaptive or specific, this box right here. And if you notice, just like we have semantic range for words, meaning that some words spill over a bit, the natural killer cell is kind of like a intermediator for both. It functions in both the innate immunity and the adaptive immunity. If you watched the first video, we talked about how natural killer cells are under the tissue, living in the tissues there, and they're kind of like on guard duty, like Marines on guard duty. They say, halt, who goes there? And you have to show your identification. If you have the wrong identification, he takes care of you. And so thank God Marines aren't like that. They're not just going to take care of you right then and there. They'll either escort you off the base or what have you. So if you're looking at the innate immunity, we have all these different cells that play roles in immunity. You have your macrophages, which are large phagocytic killers. They engulf bacteria. We have neutrophils, which also can function as a phagocyte, and they're usually the first to come on a scene once they're mobilized and told to go after something. Eosinophils, very rare. These are associated with allergies and parasites, which also can become a microphage or a macrophage. Neutrophils are usually called a microphage because they're smaller. See, both of these are smaller than this one. That's why this one's called a macrophage. And then we have basophils, which are specific for in tissues, in the blood, for producing chemicals. And then here's a dendritic cell, which could be many different types of cells. And a dendritic cell is an important player in the immune system, is in that they can actually tag a bacteria or virus and bring it to these guys. Let's say these guys are living in a lymph node, the T cells and the B cells, bringing it to them, and it's going to present the antigen to them. Now, natural killer cells are functioning in both, as we'll see. So specific means it's very specific, and adaptive, it's trying to adapt to what environment you're in. So if you're in an environment like the planet Earth, You have to adapt to specific viruses that have gone everywhere. The respiratory virus or a respiratory droplet type virus spreads everywhere. To try to say that it's not going to spread everywhere is ludicrous. It's almost an impossibility. The common cold's been everywhere. That's how it spreads, the same way. The flu has been everywhere. Whether you get really sick with the common cold or really sick with the flu is all dependent upon many different factors. One is the host, meaning you. How healthy is the host? And so that's another topic. So specific or adaptive immunity is the ability for the body to defend against specific bacteria or toxins or viruses or foreign tissues. And it has two properties that distinguish adaptive from the innate. Remember, the innate wasn't very specific. And there was many different things associated with the innate, including your skin, your mucous membranes, your chemicals, proteins, and so on. But in the adaptive or specific immunity, we have the specificity of foreign antigens and identification of identifying these foreign antigens. So we have to be specific on what the antigen is you're going to go after. What's the target? Who's the target? What's the target? You need to know. And also, these cells that are going to be made to go after targets should know self from non-self. Meaning, if you're making a whole bunch of cells that are killer cells, you don't want them to kill yourself, your normal cells. So they have to be filtered through. And we'll talk about that in another video. So this one here, we also, whenever you're making these cells to go after these foreign guys, so you're going to build up your troops, right? You also have to have memory cells so that the second counter with that antigen will prompt an even swifter, more vigorous immune response. So the terms, we're going to go over a couple of terms here. One term is antigen. Of course, you must know that by now, that it's a substance that are recognized as foreign, and they can provoke an immune response. And so, like on guard duty, if you have a person from the other side of the army or another army coming in who's your enemy, that wouldn't be good to just let them come in the gate and just do whatever they want to do. You have to be able to identify who the bad guy is, right? So an antigen is something that would be foreign to the human body and could provoke an immune response. Immunology is the branch of science that deals with responses of the body when challenged by antigens. So it's the study of how your body does this. So we talked about the lymphatic system, we talked about the blood system, the circulatory system, and it's not, those two alone are not the immune system. The immune system is really multifactorial, as you saw in the innate immune system, your skin, your mucous membranes, there are proteins circulating, chemicals, all those different chemicals we talked about. We have the complement system. We have all these different safeguards prior to a cell breaking through the tissue and invading your tissue even further. And once it invades the tissue, we even have guards on guard duty like the natural killer cells and other macrophages. But then we have this. We have this specific immunity or adaptive immunity that can now program to make new warriors, so to speak, and helpers designed to go after specific enemies. So lymphocytes are, we know what they are, they're white blood cells, right? So you have white blood cells, leuco means white, and there's many different types of white blood cells. We have the granulocytes, the agranulocytes, we learned all that. And then you have these lymphocytes, which are specific types of cells, and lymphocytes include B cells, T cells, natural killer cells, and macrophages. And so there's two basic types involved in specific immunity are the B cells and the T cells. So that's what makes specific or adaptive immunity different from the others, because macrophages are involved in innate immunity, and so are other macrophages. You know, we have natural killer cells that are involved in innate and adaptive. So the thing that makes this adaptive immune system different is the B cells and the T cells. And so let's talk about them for a minute. All blood cells, including B and T cells, come from bone marrow. The B cells mature within the bone marrow, whereas the T cells will mature in the thymus. Before a T cell can leave the thymus, they must develop what's called immunocompetence. That means the T cell must have the ability to make very distinctive proteins to be inserted on their cell membranes, and when they leave, these proteins will be used to fix as antigen receptors. So those proteins will be the guys who are going to grab the antigen, the bad guy, so to speak. Also, you want to be able to identify self from non-self. That's all part of immunocompetence too. So when you make these new T cells, they better not be attacking the human body, right? So we have to make sure that they can do that before they leave. And there are two major types of T cells involved in this system, and that's the T helper, identified by the protein CD4 in the cell membrane. So sometimes they call it CD4, T helpers, the same. And then the T cytotoxic cells identified with the CD8 protein. Sometimes they call cytotoxic cells T killer cells. So two types of adaptive or specific immunity. You have cell-mediated and humoral. Okay, so here we go. We're talking about this system that's very specific, and there's two basic branches. One is the cell-to-cell combat guys, and that would be your T cells. The other is the humoral or antibody-mediated, and that has to do with your B cells. Not that T cells don't have a function in both, they do, but for the most part, the T killer cells are the ones that are going to go after, combat, and attack a cell. The humoral or antibody cells, or antibody mediated, is B cells that are going to produce, eventually they're going to turn into plasma cells and manufacture and produce many antibodies. So cell-mediated cell-to-cell combat. These are cytotoxic cells, CD8 cells, that directly attack invading antigens. They're most effective for intracellular pathogens, such as virus, bacteria, fungi inside a cell, and even some cancer cells and some foreign transplanted cell. So what they're going to do, they're going to attack a cell that's been infected, so to speak, inside. So they're looking for some sign that tells me that that cell's infected, and it's going to go after it. So the cell-mediated or adaptive immunity always involves cells attacking cells. The T helper cells, CD4, aid in both cell-mediated and when we talk about the B cells and the antibody-mediated. Okay, here we go. Antibody-mediated B cells will transform into plasma cells. It's a mature B cell, basically. And when they do transform, they are going to synthesize and secrete antibodies. Another name for antibodies are immunoglobulins. These antibodies can bind to and inactivate a given antigen. This works mainly against the extracellular pathogens. So this works with stuff that's outside of the cell that's floating around, such as viruses, bacteria, fungi that are in your body fluids. Both systems, cell-mediated and antibody-mediated, work together to eliminate large numbers of copies of a particular antigen. So before we talk about this, we need to understand major histocompatibility a little better. And major histocompatibility is basically the cell identity marker of a cell. In other words, on the cell membrane, there'll be proteins, usually a glycoprotein, that identifies you as yourself. And those proteins have a very specific shape that are only unique to you. Nobody else on the planet has the exact same as you. And each antigen, each bacteria, virus has their own proteins on it. And so there's two basic types. The first type we call MHC1, and that's the molecules embedded on the plasma membrane of all cells. And it's usually a glycoprotein. MHC2 appear on the surface only of APCs. Now an APC, what is an APC? An antigen presenting cell. They could be cells that were dendritic cells, like Langerhans cells, a macrophage, and even B cells found in the epidermis, dermis, and mucous membranes that can incorporate the MHC of an antigen on its membrane. on the surface of it to present it to immune cells like the T cells and the B cells. So before we go on, this word dendritic cells always drove me crazy. And a lot of books don't clarify it very well. But basically what it is, they're named for this shape. They have these like pointy things coming off like this. That's why they call it dendritic. They look like dendrites of a neuron. And they're responsible for the initiation of the adaptive immune response, and hence function as the sentinels of the immune system. Dr. Paul Langerhans first described dendritic cells in human skin. Those are the Langerhans cells. He thought they were nerve cells at first because of the dendrites. But right underneath your skin you have these. So when something tries to get in, they're automatically going to engulf it. They're going to be a phagocyte. So here, a bacterium is engulfed by phagocytosis into a dendritic cell and encased as a phagosome. So here's the bacteria. It has specific proteins on it that he identified this guy as a bad guy. As soon as he realized he was a bad guy, he engulfed it through phagocytosis. So it brings it into the cell and it fuses with lysosomes and forms a phagosome, and it digests the bacteria, breaks it up into little tiny pieces. And now those tiny pieces sometimes escape, as we talked about before, and then it's going to take these proteins and incorporate that exact shaped protein on its membrane. And now that is MHC2. So this was his MHC1, these little red proteins for that bacteria had his own MHC1. Then he engulfed the whole entire bacteria and stripped off those proteins and incorporated on his own cell membrane as a MHC2. This is the one he's going to present to a T cell and a B cell that's found in the plasma and found in the lymph nodes or lymphatic tissue. So now we have different types of antigens. Some are found inside the cell. The cell's been infected, and we have some that are found outside the cell in between cells. And the term endo, the prefix endo, means to enter, right? Like endoscopy, you do an endoscopic exam, you enter someone's throat, right? Or whatever. So now we have the endogenous antigens, meaning these are foreign things that are found inside a cell. Now notice, this is a cell and was infected with this virus. And then it's going to take those viral proteins and it's going to place it as an MHC2 here and also over here. Two different target spots. And so these proteins were the viral proteins, and they put it on here as an MHC2. So endogenous antigens means it's within the cells. These are antigens found within cells, such as viral proteins, toxins from intracellular bacteria, abnormal proteins produced from a cancer cell, and most body cells can process and present endogenous antigens. So this guy got infected and he's presenting it. The display of the endogenous antigen bound to the MHC1, or MHC2 as I called it here, signals that this cell has been infected and needs help. Well, sorry to say, the help he's going to get, he's going to be destroyed. But before that, someone's going to take these antigens and process it and make more warriors to go out after the same proteins, such as that specific virus. And that's what happens with the B cells and the T cells. Right? Here's the exogenous antigen. So an exogenous antigen is something outside the cell. Exo means to exit, right? Exit. Exogenous or exogenous antigen is something that's foreign, but it's outside the cells. So here is a phagocytic cell, or probably a macrophage or a microphage, and they're found floating. It sees it and it engulfs it. Okay. So now he's going to become an antigen presenting cell. And here's the antigen from that bacteria that he processed and incorporated on the outside of his membrane as MHC2. So now he's using that to present that to the T cells and the B cells. And this is effective with bacteria, bacterial toxins, even parasitic worms, inhaled pollens, viruses that have not yet infected a cell. Nice to have that, right? Here's another discussion about antigen-presenting cells. There are specialized cells that process and present exogenous antigens. They could have been a dendritic cell like we talked about, a macrophage, a B cell. It could be found in your epidermis, your dermis, your mucous membranes. So as someone coughs on you, it goes into your nasal mucous membrane, and you have these cells living in the mucous membrane. And then what they're going to do, they're going to take that antigen, that protein, from that antigen. So here it is. This is an antigen uptake on an immature APC. It takes the antigen in. It's going to process it, incorporate it on its outside membrane. Okay, so the APC now migrates to the lymph tissue and presents it to the T cells. Now you have two types of T cells, CD4 and the CD8, or the T helper cell or the T killer or cytotoxic cell. And so it's two different types of membrane receptors, and they membrane incorporated in two different spots, and it's going to affect this T cell receptor here and this T cell receptor here on this guy. So once the T cell binds to the MHC2 complex of the APC, it triggers the adaptive immune response. What is that? It's going to inform T cells that that is the specific intruder that's present. And then it's going to go to work and start making new T cells that are programmed for that. And then it can also make B cells too. Let's look at this. So let's talk about cytokines first. We have to get through some of these topics a little more. So we've talked about cytokines before. They're hormone-like substances that have a very short half-life. They have autocrine and paracrine-like function. They can either stimulate or inhibit cell function. Cytokines are intercellular mediators, or the way immune cells communicate to each other. You can abbreviate, the interleukins are a type of immune type of cytokine, and sometimes you number it IL. So you can have IL and a number. And they're named, the numerical number was based upon when they were originally discovered. So I'll give you an example. Interleukin 1 is produced by macrophages and promote proliferation of T helper cells. It can also act on the hypothalamus to induce fever. Interleukin 2 are secreted by T helper cells and T cytotoxic cells and B cells, and it can activate natural killer cells. Interleukin 4 is also known as B cell stimulation factor, and it's produced by T cells, and it's a co-stimulator for B cells. It causes B cells to mature into the plasma cells and secrete the immunoglobulins, IgE, specific immunoglobulin. It also helps to promote growth of T cells. Then we have other chemicals that are cytokine-like chemicals. Tumor necrosis factor, produced by macrophages. It's a membrane-bound chemical that stimulates the accumulation of neutrophils and macrophages at the site of inflammation. That's good, right? And then macrophage migration inhibiting factor, it's produced by cytotoxic cells, and it keeps the macrophages from leaving the infection site. So it makes them stay there for a while so they do their job. So we have other chemicals that are called chemokines. They're the ones that are like signaling the movement of a leukocyte. So they're the ones associated with chemotaxis when we talked about chemotaxis, which will attract, so to speak, neutrophils to the site of infection. Interferons are other chemicals that are produced by virally infected cells. We've talked about this before, and they can help prevent non-infected cells from getting the virus and spreading it. Okay, so cell-mediated immunity and activation of T cells. Let's talk about that for a few minutes. So cell-mediated immune response starts with an antigen. It's the antigen of an infected cell, let's say. So here's a cell that's infected. This macrophage engulfs that infected cell, incorporates that protein as an antigen MHC complex. Then your T cells will bind to that. And now notice how the T helper cell is going to release cytokines that are going to trigger and activate the memory T cells to make memory cells, and then also make clones to go after that specific bacteria. and send natural T killer cells after it. And so we have this antigen of an infected cell, and it's engulfed by a phagocyte. It incorporates that antigen, presents it to the T cells. The T cells are involved, and they activate each other. So the T helper cell can help activate the T8 cell, and then the activated T cells undergo clonal selection. And this results in the formation of clone cells that recognize the same antigen as the original T cell, along with the T memory cells. So now you're going to have a whole bunch of these clones that are all programmed to go after this. But then you'll have memory cells stored in your memory, in your body, that can become active again the next time you're infected. It's important to note that most T cells are generally inactive, and they have to be activated. So T cells have these T cell receptors, and they recognize and bind with those specific antigens, just like we saw. And so they take them in the form of an antigen MHC complex. We saw that. And there's millions of different T cell receptors that recognize specific antigens or specific MHC complexes. Antigen recognition also involves CD4 or T helper cells and CD8 cells, or the T killer cells, or cytotoxic cells. And they interact with those MHC antigens and help maintain the T cell receptor MHC coupling. They refer to these CD4 and CD8 proteins as co-receptors. Then we realize that antigen recognition by T cell receptors with CD4 or CD8 is the first signal to activate T cells. Then co-stimulation by cytokines such as interleukin-2 and as many as 20 other different co-stimulants enable the antigen and the T cell to adhere to each other for a period of time. Recognition and co-stimulation are necessary for the immune response. Okay, so recognition without co-stimulation leads to anergy. That's a state of inactivity of T cells and B cells. We don't want that, right? We need to have that co-stimulation there. Okay, CD4 or T cells, T helper cells, excuse me. How do they get activated? Activation and clonal selection of T helper cells. The cells with the CD4 protein develop into T helper cells that recognize exogenous, remember exogenous outside, and fragments with the MHC2 at the surface of them. The T cell recognizes exogenous antigens, a fragment that's incorporated on the APC cell with the aid of the CD4 protein. The T helper cells bind to the APC. Co-stimulation occurs, activating the T helper cell. The T helper cell undergoes clonal selection and forms clones of the T helper cells and makes memory cells. So look, here you have a bacterial antigen being engulfed by a dendritic cell, incorporating the class 2 MHC complex on its surface with, that's identifying this bad guy. And then it binds with the CD4 protein of the T helper cell, CD4. And then that's going to trigger it to make clones. And also, there's co-stimulating molecules that are going to be affected. So without the co-stimulation, this wouldn't happen. And then you're going to make T helper cells that now are programmed for that specific bacteria. And you're going to have memory cells that only get activated later. So look, within hours, the T helper cells secrete a variety of cytokines. And those cytokines will be working on the T8 cells or the T killer cells to activate them. And so the memory T helper cells will only be activated the second time it's exposed to the same antigen. But that response will be much quicker and more effective. T helper cell co-stimulating a T cytotoxic cell. So now that T4 cell that was activated already, as we said, can make a lot of cytokines. And so now the CD8 cells are recognizing the foreign antigens combined with the MHC1 on the T4 cell. So here we go. Here's an infected body cell. and here's a T helper cell, right? Releasing and co-stimulating this T cytotoxic cell that comes into contact with that MHC and that protein of that antigen. So the T helper cell is going to co-stimulate the T8 cell. The T8 cell is going to bond with that APC MHC complex. It's going to be now an activated T cell, T killer cell, or T cytotoxic cell. It's going to make clones of itself, and these clones are active and they are programmed to go after that specific protein. And then you'll have memory cells that will only react later on. And so these are important in body cells infected by microbes, some tumor cells, cells of tissue transplantation. Recognition requires the T cell receptor and the CD8 protein coupling with MHC1 complex, and then co-stimulation by interleukin-2 from the CD4. There it is. And the cytokines produced by other CD8 cells can help as well. And so once activated, the CD8 or killer cell is going to undergo clonal selection and form memory cells as well, which will respond a second time exposed. So you have the clonal cells produced to go out and go after that protein of that bacteria, and then you have the memory cells that can be functioning later on if you get exposed again. So let's talk about how a cytotoxic T cell does attack. So here, cytotoxic T cells, or CD8 cells, or T killer cells, attack other cells and release granzymes. They trigger apoptosis, and they make perforins that trigger cytolysis. We've talked about this already, right? So they also are your immunological surveillance cells, and working together with macrophages and natural killer cells, the CD8 cells that are already programmed are going to be surveying to see if that one specific bacteria is out there. And they also can recognize cancer cells, virus-infected cells as well. So here is a T helper cell, okay? And this is a pathogen of a phagocyte being engulfed and incorporating on its surface as an MHC. The T helper cell takes that on and now becomes an activated T helper cell. It's going to release cytokines, which can activate T cytotoxic cells. And as it activates a C cytotoxic cell, it will go after that protein of that same protein, and it's going to attack it and destroy it. So let's talk about humoral or antibody mediated immunity. So this is when we start talking about B cells, and they usually stay put where they're at, and then they're activated in the lymph tissue, such as the spleen, lymph nodes, or mucus-associated lymphatic tissue of the GI tract or wherever. Then they can become activated and undergo clonal selection. And of course, just like the T cells, they're going to make a whole bunch of new clones of the B cells, right? And first of all, the antigen is taken into the B cell, processed, and moved to the cell membrane as an MHC2 complex. So here's the B cell, here's the antigen, taken into the cell, and it incorporated this MHC2, right? Okay, so it's recognizing it, and the complex needed, MHC2 delivers needed cytokines, and that's what's really going to activate. The cytokines are the co-stimulators that trigger the proliferation, differentiation, and clonal selection of plasma cells and memory cells. Now, the plasma cells, like I said before, are the mature B cells, and they're going to secrete millions of antibodies a few days after exposure, secreting them up to 4 to 5 days until the plasma cell dies. All of the B cells of a particular clone secrete only one type of antibody. It's identical to the original antigen that the B cell first responded to. So the B cell's responding, you have an activated B cell, and it's activated because of cytokines from the T helper cell, and then you get this clonal production of plasma cells, which are releasing. These are the antibodies they're releasing. So it secretes a lot of antibodies, and those antibodies are going to be used to tag the bad guy. And then you have the memory cells that go into memory that can mobilize the next time you're exposed. So here's what an antibody looks like. Antibodies will eventually form an antigen-antibody complex, because what happens, they will attach to the antigen. So here's an antigen right here attaching to this antibody. And this is just like a schematic representation of an antibody, and here's like a more graphic view of what an antibody looks like with all those amino acids and globular protein here. And so if you look, antibodies have the ability to combine specifically with the epitope of an antigen, a portion of the antigen that sticks out, or a portion of the antigen itself. And so that could have been broken off as a piece, and now it can bind to that. That's what an epitope means, something that came off of it. And it triggers their production. The antibody structure matches the antigen. Binding occurs at the antigen binding site of the V region. There it is right there. And this is the V region, of course, here, and that's the binding site, antigen binding site. So the antibodies, also known as immunoglobulins, abbreviated IgS, are glycoproteins called globulins that contain 4 polypeptide chains. 2 chains are identical and called H for heavy chains, consisting of about 450 amino acids. So if you look at the heavy chain here, there's going to be 2 of them, and it's got about 450 amino acids there. And then 2 short carbohydrate chains, and then it has 2 light chains of amino acids. So you see the light chain right here, that would have 250 amino acids. And there is also disulfide bonds holding them together. So there are different classes of antibodies or immunoglobulins. And this is a good example: IgG, IgE, IgD, IgM, IgA. You probably heard of these, immunoglobulin G or immunoglobulin E. You may have heard of these at some point. IgG is the most common, and protection from bacteria, virus by enhancing phagocytosis. Now, we're going to look at the function of an antibody in a few moments, but they can neutralize toxins. They can do several other things associated with phagocytosis, and we'll talk about it in a moment. But 80% of all immunoglobulins are this IgG. It is the immunoglobulin that crosses the placenta in babies because they can't make it their own until age 2. Really, a child cannot make enough IgG until age 2. And here's the argument for certain types of vaccines before the age of 2. Argument against them. Because if the child cannot make the immunoglobulin you're trying to get them to make, why expose them to that antigen? It doesn't make sense at all. And so, just to tell you a personal thing, my first daughter, I wasn't totally against vaccines like my friends, because I wanted to be a critical thinker, and I wanted to do the right thing for my children. I'm not going to be a bonehead and just have like a belief that's it. And so I did my research way back then, and I realized that she won't be making her own IgG. So any vaccine that's going to try to mobilize immunoglobulin G, I'm going to postpone until age 2. So I didn't have her vaccinated until age 2. And she was a very healthy kid. Anyhow, IgA is about 10 to 15%, and it's mainly found in breast milk, sweat, tears, saliva, mucus, and the GI secretions. And stress will lower your levels of this. So stress is not a good thing for your immune system. But it does provide protection upon the mucous membranes against bacteria and virus. You want that, right? So reduce your stress if you don't want to get a virus. IgM is about 5 to 10%. It's the first antibody secreted by plasma cells after the initial exposure to any antigen. It activates complement and causes agglutination, which is one of the activities that antibodies do, and lysis of microbes. It can also circulate as an anti-A and anti-B in your blood typing. So when you're looking at an anti-A immunoglobulin that's going to attack the wrong type of blood. Let's say you're an A type and someone gives you a transfusion of B type, that's the one. IgM is the one that's attacking that other blood type that you're not supposed to be getting. IgG is only 0.2%, and it's found on the surface of B cells, and it's involved in activation of B cells. IgE, look, it's less than 0.1%. That's really low. But it's found on mast cells, basophils, and involved in allergic hypersensitivity, and can provide protection against parasites. One of the things you would look for is an elevated IgE in a person who might have an allergy. Okay, so here's how antibodies function. And this is like, there's a few more functions, but this is the most important ones. Neutralization. The antibodies will block specific sites on the antigen and prevent them from binding to receptors on tissues. They can agglutinate. Antibodies bind to more than one antigen, creating cross-linked antibody-antigen complexes that are so large it causes clumping. Precipitation, it can actually go after soluble molecules and do the same thing and cause them to precipitate out. And antibodies can use opsonization and positive feedback mechanism to promote phagocytosis by helping complement fixation. And then antibody-dependent cell-mediated cytotoxicity. That's a mouthful, huh? ADCC. And that's the process of killing antibodies that are coated by specific leukocytes, like natural killer cells and eosinophils. So in other words, it will coat the specific antigen. Antibody will coat it, and then these guys will target it.